Hello, welcome to Chapter 47, Animal Reproduction and Development. By the end of this chapter, you should be able to contrast asexual and sexual reproduction and describe the structure of sperm and eggs, contrast external and internal fertilization, describe the events of fertilization in animals, including the mechanisms that prevent fertilization by more than one sperm, describe the events of cleavage during animal development, describe the collective and coordinated cell movements that lead to each distinct cell layer and body axis, and the body axes during gastrulation, and describe the processes that form the somites and the neural tube. Alright, so let's just remind ourselves about what asexual and sexual reproduction are. So asexual reproduction is reproduction that is based on mitosis, meaning you're just making, more cells like the cells you already have by dividing. And asexual reproduction occurs without the fusion of gametes. So if you have offspring by asexual reproduction, they are genetically identical to each other and also to the parent that made them. And we have plenty of kinds of animals and plants in which individuals can clone themselves, making large numbers of identical copies of themselves. So if you've ever seen a dandelion make those like white puffy things and then all the seeds go everywhere, those are actually clones of the parent dandelion. Alright, sexual reproduction is based on meiosis and fusion of gametes, and so if you undergo sexual reproduction, the offspring are going to be genetically different from each other and also genetically different from their parents. So I don't think we've talked about this before, but let's review how in animals sperm and eggs are made. Okay, so spermatogenesis just refers to making sperm. So you start out with an initial cell, which is this is the lineage of cells that gives rise to sperm. So the progenitor cell that does this is the spermatogonium, and so it might have to divide by mitosis to make more spermatogonia, right? And then within this cell you have meiosis happening. So meiosis occurs. So you have two copies of each chromosome, right? So those duplicate themselves and then divide once for meiosis 1. So now you have in each of the cells, if this is chromosome 1, you have chromosome 1 from your mom over here maybe, and from your dad over here, and then here's chromosome 2, Alright, so that's meiosis 1. Then in meiosis 1, each of the two identical copies of the chromosome 1 from your mom, or the chromosome 1 from your dad, or the chromosome 2 from your mom or your dad get split so that each of the cells now has a chromosome made up of one chromatid, right? So the name of the cells after meiosis 1 are the secondary spermatocytes, and then after meiosis 2, they're called spermatids. I will not ask you the vocabulary on this slide particularly, just be aware that the final product of spermatogenesis is going to be 4 mature sperm. This is not quite the same as what happens with eggs. So we call the production of eggs oogenesis. So you start out with a diploid cell, and it undergoes mitosis, differentiation, and chromosome replication to make the primary oocyte, and then that undergoes meiosis 1. And at this point, you produce one kind of large cell, that's the secondary oocyte, and then a smaller one called the polar body. And then when the secondary oocyte divides, it produces a larger ootid, and then this little polar body, and then the polar body over here produces these other two polar bodies. So basically what you have here is one viable cell that could actually go on to become an egg, right? And then these three little nubbins of cells that don't really have cytoplasm, and they will never be fertilized, so they're not going to become offspring, okay? So in oogenesis, if you start out with one cell, instead of ending up with 4 gametes or 4 eggs, you only end up with 1 egg, okay? So one cell results in 1 egg. In spermatogenesis, 1 cell results in 4 sperm, okay? That is the main thing. Do not bother and go and memorize ootids and secondary oocytes and primary oocytes and the oogonium or whatever, okay? Okay. Let's talk about the structure and function of eggs. So if you have a mature mammal cell, mature mammal egg cell, it should have the things you would expect, like a cell membrane. Should also have a haploid nucleus. right? So that nucleus is haploid because it's a gamete, it's an egg cell, right? It should have all of the other organelles that you would expect. Oop, not here. All of the organelles that you would expect inside the cell. But the other thing is that it should have a yolk with fats and proteins that are going to be used to nourish the offspring. All right, on the outside of the cell membrane is a glycoprotein vitelline envelope, okay? So we can see here, this is a sea urchin. So here's the vitelline envelope right here, okay? And in sea animals, on the outside of the vitelline envelope, you have this jelly layer because they're kind of in salt water, so they need extra layers of protection. And then you have the plasma membrane that is on the inside here. So you have your membrane, and then on the outside of that, you have your vitelline envelope. And so the sperm has to come in here and make contact with the plasma membrane on the inside. And when that happens, that initiates fertilization. And so that's important. Other animals have amniotic sacs with these 4 membrane-bound sacs. Other animals produce amniotic eggs with the 4 membrane-bound sacs. So remember we talked about that when we talked about animals with the amniotes. So this is a very simple egg because it is the egg of a sea urchin, okay, not an amniote. If you look at human egg cells, you can see that they have more fancy stuff going on the outside. So the vitelline envelope, which you can see is just this dark blue layer over here in the sea urchin, is actually thickened in an area called the zona pellucida right here. Okay, so it's this thicker zona pellucida, and on the outside of that there's a layer of cells called the corona radiata, and so the sperm have to get through the corona radiata. Otherwise, we have pretty much the same things in here. So here's your plasma membrane on the cell. Inside of the vitelline envelope, which is in this case called the zona pellucida, we have the cytoplasm of the cell. We have little bits of yolk here. And we have those polar bodies, remember? The polar bodies stay stuck to the egg cell, okay? All right, so that's what we got going on in these eggs. So there's a lot of variation in fertilization and egg development amongst the animals, but let's just make sure we got the vocabulary here. Fertilization is the joining of a sperm and an egg to form a diploid zygote, okay? So if you have external fertilization, it means that individuals are releasing their gametes into the environment, like the sea star is releasing eggs into the seawater. And often there'll be kind of pheromonal control of gamete release, so that you have like all of the sea urchins of a particular species releasing their sperm and eggs at the same time, and so that helps the sperm and the eggs of the same species to find each other. You have internal fertilization in most terrestrial animals and in a significant number of aquatic animals, and so that basically means that the males aren't just like putting sperm into the water and females putting sperm into the water. Instead, males have to deposit sperm directly into the female reproductive tract through copulation. So copulation just refers to the process by which the male kind of gets the sperm into the female. So males might be packaging their sperm into a spermatophore, like we talked about in chapter, I don't know what it was, chapter 39. And then the spermatophore is then placed in the female's reproductive tract by the male, or by the female in some species. So in this species, the male is kind of on the bottom here with the spermatophore, and he will kind of do this fancy dance where he wiggles his butt around, and he will manage to work the kind of stem of the spermatophore into the female's genital opening so that the sperm can be transferred. All right, so sperm competition can sometimes happen. So this is competition between sperm from different males to fertilize a female's egg. So this is a form of sexual selection, where males are competing for the opportunity to reproduce with a female, right? And this happens in species where females will mate with many males. And so if you look at species that mate with many males versus species in which females and males mate monogamously, males tend to produce much larger numbers of sperm. And so this is because each male is trying to make like the most sperm so that it can outcompete the sperm of other males that have also mated with the same female. You also have males with genital adaptations to increase fertilization success. So This is a Calabrosis maculatus beetle, and it has these long genital spines, and so the male will put his genitalia inside the female and use that as kind of a scrub brush to get out the sperm of the previous male that has mated with the female. And so we know that this is a trait that is sexually selected because males that have longer genital spines father more offspring. So we know that there's variation in this trait, and variation in this trait is associated with greater mating success in males. And so you have kind of a female component as well, in which females may be doing choosing. So in some species, females will choose which male is going to fertilize her eggs by the female manipulating sperm storage, or sometimes females will physically eject sperm from undesirable males. And so this is called cryptic female choice. And that is because the selection of sperm by females is hidden from males, or potentially it could have been hidden by the original male researchers who were looking into this issue and they couldn't figure out whether females were doing this or not. In the cricket example I showed you a few minutes ago, females will often exhibit this kind of cryptic female choice, although it's not very cryptic. If a female doesn't like a male, she will just take his sperm packet, which is kind of sticking out of the back of her body, and just take it out, and then the sperm won't transfer. All right, so let's talk about the cell biology of fertilization. So we're using the sea urchin egg and sperm system, because this is a very common research system for studying fertilization. And so we know that the way that the sperm get to the egg in the water, out of all of the rest of the water, the way that the sperm find the egg is that the jelly of the egg secretes a chemical, and the sperm follow a gradient of chemical, meaning they go in the direction where there's more of this chemical, and the place where there's more chemical is right near the egg. Okay? So the first thing is that chemical signals are attracting the sperm to the egg. Next, the head of the sperm binds to the jelly layer. Okay? When the sperm bind to the jelly layer, that triggers something called the acrosome reaction. And the acrosome reaction causes the contents of the sperm's acrosome to be released. So you can see here, this is the acrosome reaction, and then the bit at the tip here of the sperm is released into the jelly layer. All right. Then the enzymes released, then the enzymes that are in the acrosome release digested portions of the jelly layer and the vitelline envelope. Okay. I feel like that's not worded very well. The enzymes are digesting part of the jelly layer and vitelline envelope. Okay. Also, when the sperm contacts the egg, that causes the sperm to start wiggling its flagella really vigorously, which pushes the sperm through the jelly layer to where the plasma membrane is. All right, once the sperm gets all the way through the egg's plasma membrane, okay, there are specific proteins in the plasma membrane of the egg and the sperm that induce the membrane of the egg and the sperm to fuse with each other. So when the membranes fuse with each other, that allows the nucleus, or the contents of the sperm, to be dumped into the egg. Everything got messed up here. But basically what it's saying is that, you have the entry of the sperm nucleus into the egg, and then, I don't know if it says it here, but relatively soon after this happens, the nucleus of the egg and the sperm will fuse with each other, and then fertilization is complete. All right. So here we have a reporter from the Fertilization channel. Let's call her Mindy. Mindy has some questions for you.
Yes, I have some questions for you. How does a sea urchin's sperm find the egg?
Who's that? Oh, I made up that voice myself.
Oh, okay. That's creepy. Don't do that again.
Okay, sorry, dog. All right, well, I'll ask you. As Mindy, how does a sea urchin's sperm find the egg?
Oh, it's following the chemical gradients that's being secreted by the jelly.
Yeah, that's true.
That's how I find peanut butter and jelly sandwiches. I follow the chemical gradient to the jelly.
Okay, good. So when the sperm binds, what happens? It releases the acrosome layer and Yeah, and then it causes the acrosome reaction, and then the acrosome like digests the jelly a little. Yeah? So how does the sperm get through the jelly layer and the vitelline envelope?
Oh, it's like from the acrosome reaction, it's digesting stuff, and also the sperm starts whipping its tail around.
So what causes the sperm's flagellum to start beating?
I think it's contact with the membrane.
All right? So what causes the sperm and the egg membranes to fuse?
That's just when the sperm gets all the way up to the membrane.
Yeah. Okay. All right. So, if you think about it, the ocean is filled with a billion bajillion sperm and eggs of all sorts of different sea creatures.
Now I am never going to be able to enjoy the ocean again.
Really? You eat anything. Why would you care?
Okay, I guess I'm just being dramatic.
So this causes a problem, right? So gametes of the different species are all kind of in there together. So there has to be something that causes the sperm and the egg of the same species to recognize each other and to reject the other species. So there's a protein known as bindin, which is found in the head of the sperm, and it binds to the plasma membrane of sea urchin eggs in a species-specific manner, meaning it only binds to the plasma membrane of its own species. And so if you look at proteins from very closely related species, they have very different proteins. And the way that this works is that the egg plasma membranes have receptors, and they only bind bindin from sperm of the same species. All right, so why does an egg prevent the fusion of more than one sperm? Is it because the egg's jelly coating secretes an attractive chemical?
No.
Is it because egg cells are massively larger in volume than sperm cells?
No.
Is it because cell fusion is an essential component of fertilization? No. Is it because sperm and egg cell genomes are haploid?
Wait, what does that have to do with anything?
Okay, no. And is it because embryos with more than 2 sets of chromosomes cannot develop properly?
Oh, it's that one.
Yeah. All right, so what prevents more than one sperm from entering the egg? Meaning, what prevents polyspermy? Well, we can look here and see that, so you have a sperm nucleus that has just entered an egg here. So it got all the way through the fertilization process, and it's just dumped its nucleus into this egg, okay? So immediately after sperm-egg contact, the membrane of the egg is depolarized. So you have a wave of calcium ions that are released from the starting point. And yeah, so calcium starts to be released where the sperm nucleus enters the egg and it spreads all the way around. And while that is happening, additional sperm can't fuse. So basically the wave of calcium caused by the initial fertilization event prevent any other sperm from fusing. Yeah, so sperm entering the nucleus cause calcium ions to be released. That digests the sperm receptor, okay? And so when the calcium ions digest the sperm receptor, then new sperm can't bind, right? Because the bindin can't bind to anything. You also have water that diffuses into the space between the plasma membrane and the vitelline envelope, okay? So what we have here is, this is an egg cell, and all these little fuzzy things are different kinds of sperm, or different sperm that are attempting to fertilize the egg. Now, one of them has a head start on the other, just probabilistically, right? And so one of them wins and enters the nucleus, and when that happens, so I guess it was one that's right up at the top here, that causes the vitelline envelope to kind of fill with water and lift up away from the cell. And that forms a tough fertilization envelope that makes it so the other sperm can't get in. So this would be something that you have in a sea urchin. You do not have this exact mechanism in mammals, but we do have that calcium-mediated enzyme release, which does break down the sperm receptor protein so that you can't have a second sperm fertilize the egg after the first one has. All right, I'm going to try this video here. Let's see if it works. All right. So what you're going to see is a sperm, and I think it fertilizes around here. Okay, so the sperm that fertilizes sets off that calcium wave that goes around the egg, and it also causes the vitelline envelope to raise up. So you can see the vitelline envelope form around the outside afterwards. Oh, there it goes. Okay, so let's talk about what happens next. So fertilization will then activate development, in which you have one cell that becomes two cells, and two cells become four cells, etc., etc. So you have your all right, next you have embryonic development, and so you have your fertilized egg here with the vitelline envelope, and it divides in half, so now you have two cells and four cells, and then many cells, right? So the stage in which you have this rapid cell division is called cleavage, because the cells are cleaving from one another. And then you have a mass of dividing cells, so that's called a blastula. And as the cleavage nears completion, you have a slowdown in the cell division, and you start to have the cells migrate to the outer edge, and then it becomes a hollow ball of cells. And then you have what's called gastrulation, and that's when you have, remember we talked about that a little bit earlier when we talked about the blastopore and deuterostomes and protostomes. So this is when you start to have a kind of a dimple forming that forms the mouth or the anus. That's gastrulation. And we say that the shape of this cell is the gastrula once it gastrulates. Okay, so this is gastrulation in a frog embryo, and I think there's more detail here than we're going to really deal with in this class, but you can kind of see that you have different regions of the frog blastula, and I think you talked about this on 1113, or at least when I taught 1113 I taught this, that you have chemicals called cytoplasmic determinants that will direct which parts of a developing embryo will become which parts of the body. So the cytoplasmic determinants will determine which end is the head and which end is the butt, okay? So they will determine, which part becomes which. And then you have gastrulation that happens when you have a blastopore, that little dented bit, right? And it starts to fold inward at one end of the embryo. So here's your blastopore forming. And then the cells will go in until you get the future gut. And then you have, you start out with just the inner and the outer layer, the ectoderm and the endoderm, and then eventually the mesoderm will come in between. And, yep, and there you have the gut forming, okay? So once you have the three embryonic germ layers that are formed, you're ready for organogenesis, meaning the formation of the organs in the body. Okay, so we talked about gastrulation forms three embryonic tissue layers, ectoderm, endoderm, and mesoderm, and so these give rise to the adult organs and tissues. So the ectoderm, which is shown in blue here, forms the outer covering of the body, like the skin and the nervous system. The mesoderm, which is shown in red, forms the muscle, internal organs, and connective tissues, and the endoderm will produce the inner lining of the gut and some of the organs that derive from the gut, which you can see include part of the trachea. So gastrulation is the start of the creation of the body axes, meaning, which part is going to be the head, which is going to be the butt, which is the left, and which is the right, okay? So in frogs, the blastopore becomes the anus, and the opposite side of the gut tube becomes the mouth. And so the area where the cells first move into the blastopore will become the dorsal side, and then the other side will be the ventral side. So what does dorsal mean?
That's your back.
Yeah, and what's the ventral?
That's your belly.
Yep. So what chooses which side is which? Again, we talked about this in 1113. There are regulatory molecules called cytoplasmic determinants, and so those and also interactions between the cells will cause the cells to express all the cells have the same genes, right? But those cytoplasmic determinants and also signals being sent between the cells will cause the cells to express different genes to form, the cells on the back side will form genes associated with the back. The ones on the belly will produce proteins associated with the belly. All right, so just showing you one kind of highlight of what goes on after gastrulation, which is the formation of the notochord. So gastrulation has already occurred, so you have kind of your gut forming, which is what this hole is here, okay? And then the notochord. Remember, the notochord is this flexible rod that is on the dorsal side of the animal, and we have them as embryos, but we don't have them, when we mature, right? So the notochord is going to produce these molecular signals and induce the dorsal ectoderm here to form into a neural tube. So it'll fold in like this to make a neural tube up the middle. And so that is going to give rise to the brain and the spinal cord. So that is really important. And then you have a couple of blocks of mesodermal cells on either side of the notochord that will become organized into blocks of cells called somites. And so the somites are involved in directing some of the future development of the rest of the nervous system. All right, so which of these is the correct order of events during organogenesis? What happens first?
Well, first you got the notochord.
Okay, then what?
Then it folds the ectoderm in to make a neural tube.
Yeah.
And then the somites form.
Okay, so what numbers is that?
2, and then 3, and then 4. No, 2, and then 3, then 1, then 4.
Yep. All right, so if gastrulation doesn't occur in an embryo, then what won't develop properly?
Oh, it's, wait a minute. Lots of those things. Its anterior and its posterior won't form. Then the gut's formed by gastrulation, and then the neural tube and the somites happen after gastrulation.
Yeah, so it's all of these things. All right, so hopefully you learned a lot. You learned a lot of things here about asexual and sexual reproduction, including reproductive structures and their functions, fertilization and egg development, and also embryonic development. We skipped a couple of things in this chapter. If you have interest, you can go read them, but they won't be on the exam.